Right, well, thank you for inviting me. My name's Mark Pallen. I'm kind of the Master of Ceremonies for the bacteriology stuff here. My colleagues come along and going to talk. With a 2-hour slot, it looked a big ask. And if you ask academics to speak, they will just keep talking. So we had to do something to make sure that I just didn't give a 2-hour lecture. So in fact, I'm going to give you a 20-minute talk. Then Del Benson's going to give you a 20-minute talk. Then it's going to come back to me for another 20-minute talk. And then Apoorva's going to give a 10-minute talk, and then someone else is coming to give a 10-minute. So there will be plenty of change in kind of tempo and pace and style during this.
And lots of time for discussion.
And lots of time for discussion as well. And we have these two men here who are going to keep me to time, because I can talk for England. And in fact, I was going to bring a colleague, but he can't come. But I will just emulate him and talk his talk anyway. So that's just where we're going to go with this over the next nearly 2 hours. Now, one thing I have to say where I'm coming from, some of you may have seen that there was a TV show called Goodness Gracious Me a few years ago, and there was one character in there who used to say everything was Indian. Say, old veranda, pajama, chutney, everything Indian. I'm actually a kind of greedy bacteriologist, and as far as I'm concerned, everything is bacteriology. Humans, they're just a bunch of uninteresting metazoan cells with lots of very interesting bacteria associated with them. And so I'm now going to give you an introduction to the bacterial world, just set the scene for what we're going to talk about later in the morning. So, as far as I'm concerned, life is bacterial. Yeah, it's the first approximation, this is true. Bacteria colonize every environment on this planet, from high up in the stratosphere to deep into the Earth's crust. 80% or more of life's history is bacterial. If you look back over time, they play a key role in the biosphere. So at least half of photosynthesis is bacterial. Many of the big cycles of cycling different elements, that's all done by bacteria. Bacteria are stunningly diverse in terms of their metabolism. They can live in all sorts of things, and I'll show you some examples in a moment or two. In fact, each of you is more bacterial than you are human, in the sense that you have 10 times as many bacterial cells associated with your body as you do human cells. So that puts it in perspective, if you like. But the reason we're here, I'm going to keep going on about the fact that bacteria are so important globally, but the main reason we're here, of course, is that bacteria also cause infection, and bacterial infections are important causes of human misery and death worldwide. Just to give you some evidence, I'll show you some papers, because everything we know comes from the published literature, some experiments we do ourselves. Here are a couple of papers. There's a very influential paper from 1998 where these guys tried to estimate just how many bacterial cells there were on the planet. And they came up with this stunning figure of over 10 to the power 30. Now, to put that into perspective, there are only about 10 to the power 24 stars in the entire observable universe. So about 10 to the 12 stars in each galaxy, and about 10 to the 12 galaxies. So there are 1 million times more bacterial cells on our planet than there are stars in the entire universe. So that gives you some kind of metric there. And in terms of the diversity, this other paper here looking at the kinds of diversity that we see, they've come up with some figures there that if you look at a ton of soil, you'll find over 4 million different microbial taxa in there. So, you know, many, many more times than the number of known mammals, probably more even than the number of known insects, just in 1 ton of soil from the rugby field out the back there. In fact, people are looking in all the most extreme of places. Here's an example where they looked down at the ocean crust, and they looked actually down beneath the ocean itself and into the crust, and they find bacteria living there on the basalt of the crust and making a living. And they say that actually, in this paper, they said that there are 3 to 4 orders of magnitude greater diversity in those basalt living bacteria than there are actually in the oceans above them. Here's another example where it's recognised that bacteria actually change the course of the climate of our planet. The way the surface of our planet has been shaped by bacterial processes over the years. Here was one example where there was this snowball Earth event, a global ice age, triggered by the arrival of oxygenic photosynthesis, which carried out by cyanobacteria and the release of that oxygen into the atmosphere changed the planet, changed the climate. In fact, as a greedy bacteriologist, I have to remind you that cyanobacteria, well, chloroplasts are actually just very derived cyanobacteria. All plastids, this paper shows here, and it's been known for some time, in fact, are just very modified cyanobacteria. And I wrote a paper last year, kind of a bit of an armchair philosophy thing, saying actually mitochondria are bacteria. People say, well, you know, people are very comfortable with the idea that mitochondria descended from bacteria, but one of the modern trends in taxonomics is that you don't look at groups diverging and changing in terms of form and achieving new levels of complexity. You just look at the branch order of how things diverge. So some of you may have heard that we now recognise that birds are not descended from dinosaurs. We actually say they are dinosaurs. And people who work on what you might think of dinosaurs being dinosaurs actually have to call them non-avian dinosaurs now to be taxonomically correct. Mitochondria, by the same criterion, are bacteria. And it's time we started recognising that. So when I said that you're mostly bacterial, if we take all the mitochondria into account, you know, by several orders of magnitude, you're more bacterial than you are eukaryotic. But let's move to infection. I mean, infection is obviously important. Decisive role in history, still a major cause of death and misery worldwide. So in our cosseted Western societies, we kind of get a bit complacent, but you go out into most of the world, it's infectious diseases that are actually causing most of the death and certainly most of the misery. And even in our own societies, we have public anxieties bursting up every now and then, meningitis, food poisoning, emerging infections. Within hospitals, we see what the tabloids call superbugs, multi-drug resistant organisms. So this is not a problem that's gone away. And in fact, just to warm you up and give you a break from my voice, let's just watch this trailer. This is what you should be doing this weekend. Go and watch this film, Contagion. Came out last weekend. I've watched it. It's scientifically accurate. It's chilling. It's actually very grim, but it is what could happen.
Groundbreaking, sir. Did you mention seeing anyone who was sick? Anyone on a plane at the airport?
No, she said she was jet lagged.
The average person touches their face 3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, and each other.
We're back. No, go up to your room, honey. So we have learned no treatment protocol and no vaccine this time.
You had a seizure this morning, Birmingham.
She had a history of seizures and allergies.
As of last night, there were 32 cases.
Unfortunately, she did die.
Right. So, can I go talk to her?
Mr. Unruff, your wife is dead.
What are you talking about? What happened to her? What happened to her?
Is there any way someone can weaponize the bird flu? Is that what we're looking at? Someone doesn't have to weaponize the bird flu. The birds are doing that.
Watch this. It's transmission, so we just need to know which direction.
On day one, there were 2 people, and then 4, and then 16. In 3 months, it's 1 billion. That's where we're heading.
They're calling out the National Guard. They're moving the president underground.
People will tell you. It will tip over. The truth is being kept from the world.
Cook your samples, destroy everything.
Hello. I need you to give me the names of everyone who serviced this room. It's an emergency.
You can't panic now. I'm gonna get you. I got people too, Dr. Cheater. We all do.
Don't talk to anyone. Don't touch anyone. Stay away from other people.
We're not sick.
It's figuring us out faster than we're figuring it out. It's mutated.
Okay, so that was very dramatic, but as I say, actually scientifically pretty much 95% right. I watched the film and I was quite surprised. those of you who are awake will say, hang on, there's been a non sequitur here, because that was a virus. It's actually based on two viruses, Nipah virus in terms of symptomatology, and SARS in terms of the actual epidemiology. But can such things happen with bacteria? Well, they can. If we look back into the Middle Ages, Yersinia pestis came across Europe and decimated Europe, and a third of the population of Europe was killed during this Black Death. And you might say, well, that's part of history, but interestingly, it's now part of modern science, because people have actually been able to get people who died in the Black Death, take their molars, uninterrupted molars, and get DNA out of those, and actually show that the Black Death was caused by a bacterium, the same bacterium that we recognise as causing plague today, Yersinia pestis. And just a couple of weeks ago, they actually recovered the entire genome of the Black Death Yersinia pestis. And that was quite surprising, because it does look like this was the first big wave of Yersinia pestis infection in the world, and this looks like the ancestor of all current Yersinia pestis, which was a bit of a surprise because people thought there were earlier plagues, the so-called Justinian plague, that would have been caused by Yersinia pestis as well. So this is where modern science and history and infection all come together. In fact, microbes in history have played a tremendous role. I mean, there's so many examples. Here's just one example that I just picked because we're talking about tuberculosis a bit later. This is Muhammad Ali Jinnah, founder of Pakistan, and during the 1940s he was ill with tuberculosis. Now the first speculation that you can attach to that is, well, actually if the British knew that he had tuberculosis and the Hindus knew that he had tuberculosis, maybe they would have stalled the negotiations about Pakistan. Things might have turned out different if they knew that Jinnah was going to be dead soon. That's one kind of speculation. The other one is that he only lived for a year after Pakistan gained independence. Sadly, it was just at that transition time. The first trial of a drug active against TB was in 1947, and he died in 1948. Contrast that with Nelson Mandela, who was diagnosed with TB in 1988 at the age of 70, around the same time that Jinnah was, died. He was treated successfully, and he went on to lead South Africa to becoming a multiracial democracy, and revered around the world as someone who oversaw that peaceful transition to multiracial democracy, and absolutely pivotal. And you can speculate that if Nelson Mandela had died earlier, things might have turned out differently. We might have had some horrible meltdown, the kind of Algerian equivalent. So this is where microbes have a decisive role, and our ability to treat them and our advances in treatment have an ability also to influence the course of history. Microbes are in the news all the time. You may have heard about this cholera outbreak in Haiti. Very controversial because, again, the appliance of science using genome sequencing showed that this cholera was brought to Haiti by UN peacekeepers, by Nepalese peacekeepers. That was shown by sequencing the genomes and showing that the cholera genomes from Haiti were very similar to those from Nepal. I put this one up here. This is actually a bit of a red herring. This is a BBC News report on this guy in Japan who went round to young ladies asking them for their underpants, saying that he was investigating an E. coli 157 outbreak. In fact, he was just a dirty pervert, but it gets E. coli into the news, so I kind of, it's useful for my purposes, I suppose. There's good news and there's bad news when it comes to infection. We have seen good news. It's not all bad news. We've seen MRSA rates fall, as you can see here. But on the other hand, we see bad things happening. Salmonella superbug on the rise. But the key point is that infection's still there in the news. We have problems with antibiotics. And now, again, to give you a rest from my voice and to give me a rest, I'm going to call on one of my colleagues who has just launched this antibiotic action campaign to say a few words about antibiotics and the problems we're having with antibiotics. So this is Laura Piddock, who's one of the bacteriologists here in the medical school.
Antibiotic Action is an initiative to raise awareness of the lack of antibiotics. This is important as antibiotics are cheap and easily available drugs that most of us have taken during our lives. They've been available since World War II, but before the 1950s, many people with infections died. For instance, babies and children did not reach adulthood. The use of antibiotics has led to incredible advances in medicine, from successful surgery to cancer treatments. I'm sure you are familiar with antibiotics, as you may have taken them for a chest infection. But they are also incredibly important in hospitals to treat the seriously ill. 21st century medicine is often very sophisticated, from heart transplants to knee and hip replacements to treating soldiers returning from combat zones. Infections are also very common during natural disasters such as earthquakes, tsunamis and floods, and we need antibiotics to treat people who succumb to infections during these times. However, I'm sure you are aware that there are increasing numbers of antibiotic-resistant bacteria emerging, and such strains are being spread around the world. What is less known is that there are very few new agents being developed by pharmaceutical companies, and many have stopped developing antibiotics completely. With the increasing numbers of antibiotic-resistant bacteria and the likelihood of few new agents to treat infections, this means we are faced with a scenario of untreatable infections and a return to the pre-antibiotic era, with many more people dying from bacterial infections. This is a particular concern, not just to the seriously ill, but also to the young, the elderly, and other vulnerable groups of people, such as those undergoing cancer chemotherapy. So the wonderful advances in medicine and new treatments may no longer be possible. We need new antibiotics, and this initiative, Antibiotic Action, must make everyone aware of this pending catastrophe, which is almost upon us. We need your support to re-stimulate antibiotic discovery, research and development so that new agents are available when our current drugs are no longer useful. If you are concerned about what a future with no new antibiotics will mean to you, your family and your friends, please support us and add your name to the petition on this website. Thank you.
So there we are. When you get back to your room later, go and Google Antibiotic Action and sign the petition there. It's got 1,600 signatures so far in just a few days. They're hoping to get many more thousands so they can actually get this matter discussed in Parliament. Just a little bit of didactic stuff. I know it's not supposed to be a lecture, but just let me remind you that bacteria are different from human cells. You may have learnt all the cell biology at A level and think about nuclei and stuff like that. Bacteria don't have a membrane-bound nucleus. They don't have intracellular organelles. Pointed out, mitochondria, in fact, bacteria, but inside the average bacterial cell, you don't get that, apart from perhaps ribosomes. No introns, no junk DNA. They have plasmids and bacteriophage as mobile genetic elements carrying DNA around, and that contrasts with what we see in the human genome and in human cells. Also, it's worth just pointing out there is a difference between bacteria and viruses. Viruses have to live inside cells. They obligate intracellular parasites in the jargon. They don't have their own ribosomes. In a sense, they're parasites of ribosomes of other cells. They can have DNA or RNA genomes, but they don't have both DNA and RNA in the virus particle at the same time. You have to use electron microscopy to see them. And they typically have tens to hundreds of genes in their genomes, so quite small genomes. And there is no natural kind of phylogeny where you can draw a family tree of viruses that kind of makes obvious sense. Bacteria, on the other hand, they can be parasites, but they're usually free-living. They have ribosomes, they have DNA and RNA. They're seen by the light microscope, hundreds to thousands of genes, and we can, to some degree, draw out some kind of natural phylogeny classifying bacteria. This is the standard kind of microbiology 101 approach, textbook approach. In fact, things have got a little bit muddled lately because there's a group in Marseille, headed by Didier Raoul, who's discovered these giant viruses. One of them was found initially living in the water tower in Bradford that live inside amoebae. And these are large enough to see by light microscope, where they have over 1,000 genes in their genome. And so this clear-cut distinction is perhaps not quite so clear-cut now, but for most purposes, it still holds. Now, for the rest of the morning, we're going to concentrate on two bacteria covering the whole of infection would have just been impossible. So what we're going to do is look at two research strengths in Birmingham. We look at E. coli and Mycobacterium tuberculosis. And I think I can say without fear of contradiction, these are the two most important bacteria in the world. E. coli is important. It's important because it's a model organism, it's a biotechnology workhorse, a commensal, probiotic, and a versatile pathogen. Mycobacterium tuberculosis is important because it's a major cause of death, as Dell's going to tell you in a moment. It's also got probably the most complex cell wall in nature. Some people call it E. coli in a waxy coat. It is, it has, you know, in terms of the way it works, ribosomes and an RNA polymerase and all those kind of things, you know, much is similar to E. coli, but the one thing that is very distinctive is this waxy coat. Now I'm going to stop now, but I'm just going to ask some questions, two questions of you lot, as a bit of audience participation. So how many of you do you think are carrying E. coli at the present time? Hand in the air if you think you're carrying E. coli. So everyone is carrying E. coli, that's right. I mean, certainly only 95%. There might be one person in the room that's got a Klebsiella living where E. coli would normally live or something. But generally, all of us are carrying E. coli all of the time. You get this impression from the media that E. coli is always a bad thing, you know, nursery outbreak of E. coli or something. That E. coli lives in all of us. And the pathogenic strains of E. coli are very much the minority. So how many of you have been infected with TB, Mycobacterium tuberculosis, in this room? Huh? Your auntie has it, you haven't? In fact, all of you have been infected by Mycobacterium tuberculosis, certainly according to some taxonomic conventions, and very certainly by a close relative. The BCG vaccine is in fact only a hair's breadth away from virulent M. tuberculosis, and all of you should have been vaccinated with this. And vaccination involves infection. This is a live, attenuated vaccine, so when it gets into your arm, it actually establishes a small, limited infection that stimulates the immunity. So you've all been exposed to Mycobacterium tuberculosis, albeit an attenuated form, and it's actually an attenuated form of the bovine form of M. tuberculosis. But in taxonomic terms, they really are all one species. Anyway, I think I'm going to wrap up now. Is there anyone got any comments they want or questions they want to make at the end of that part before I pass over to Del? Yeah.
I'll go to the antibiotics. Surely if you continue to try to make antibiotics to combat illness that are basically drug-resistant now, aren't we just kind of postponing what's inevitably going to happen? Because at the end of the day, like, these microorganisms have like faster evolutionary rate than we do.
Yep.
So it's kind of like we're just trying to wait.
Okay. So I think you've asked a very intelligent question there. It's one that we discussed on the train on the way in. I have some colleagues on the train where we discuss these lively issues. I discussed it recently with some colleagues at a meeting in Cambridge over the last couple of days. My personal view is, yeah, I think the era of antibiotics as we know them will come to an end. If we're lucky, we might get a few more decades. But in terms of the length of human history, you know, this antibiotic era will be quite short because we are ratcheting up resistances. And whenever we bring in new agents of which are completely artificial and never been seen in nature before, but nonetheless the bacteria become resistant to them. So I think that you're right in the sense of antibiotics as we understand them now. But that needs to be qualified by the fact that what we're doing now when we use antibiotics is very much a blunderbuss approach. We use agents that are generally very broad spectrum, much broader. They don't just target the organism we're looking at, that we want to control or treat. They actually have enormous effects on all of the bacteria in the body, in the gut, elsewhere, at the margins where the levels of the antibiotic are getting a bit low. That's the breeding ground where you're going to breed up resistance. So what we need to move towards are much smarter interventions where we actually target virulence. So if we don't need to get rid of Staph aureus and everything else by using something like vancomycin, we can actually have some smart agents, maybe active vaccination before people come in for operations, or we can give them antibodies that are very active against the Staph aureus, very specific for that organism. And I think that's probably the way it's going to go. I'm not saying antibiotic era is over yet. I hope in my lifetime we still continue to use antibiotics. But I think you're right. If you take the Barrett-Spinoza view of the kind of look at in the view of eternity, the antibiotic era is just I think a blink of the eye. Humans have been around for 200,000 years, and antibiotics will be lucky to last another 20, certainly another 200 years. I don't think we'll be doing it that way.
Any more questions?
I mean, perhaps one other point that is worth stressing also is that some of the things that we do to make people very susceptible to infection, so when we give them organ transplants or bone marrow transplants and so forth, I think we're going to get smarter and smarter in that regard as well. So the periods when people are vulnerable to infection, hopefully we can squeeze those down so that we're not quite so much of a window there. Okay. Right, well I'll pass over to Del now. All right. You're a Mac user, you know your way around.
Well, it's great being here to give you a brief sort of talk about what we do down the road in biosciences. So I've had a long interest in Mycobacterium tuberculosis. It's an area which I've sort of covered for the past 20 years or so. And really, my interests are looking at what Mark talked about earlier in terms of this unique sort of waxy coat that covers the bacteria. What I'd like to do is really give you a sort of background in terms of why tuberculosis is important and what we're trying to do. And later, Apoorva Bhatt and Luke Alderwick, who should be here, will talk about specific projects within the laboratory that we're trying to do in terms of identifying new drug targets and new correlates in terms of potential for vaccine candidates. Really, when we think of tuberculosis, it was sort of really called Mycobacterium tuberculosis by Robert Koch in 1882. But really, tuberculosis has been around for many, many centuries, many longer than we can sort of imagine. But really, there's been a sort of interest in that people have been trying to sort of date tuberculosis, and various techniques using sort of ancient DNA or chemical biomarkers, which we also have an interest in. We've been looking at sort of infection in mummies and also Neanderthal skeletons in terms of trying to date tuberculosis. And really, when we think of sort of modern sort of genomics, what we come to the conclusion is that Mycobacterium tuberculosis isn't really a single species, but is actually a complex of organisms. Obviously, we have Mycobacterium tuberculosis, which infects humans. We also have Mycobacterium bovis, which Mark mentioned earlier, which is the derivative in terms of the BCG vaccine strain. And that affects cattle. It also sometimes affects humans and other wildlife. And really, these are sort of evolved from a sort of common sort of mycobacteria. And this is probably a conservative estimate now, about 15,000 years ago. So really, when we think about Mycobacterium tuberculosis, it's a sort of evolving organism. When we think about it in terms of as an infection, really, we have our cells as the host. This is what the bacilli look like within a residing macrophage, and this is where they sort of multiply. This is what the organism looks like when we grow it on a solid agar plate in the laboratory. So what are the key issues are, when you have mycobacteria residing in a macrophage, you have an immune response. Either this will go on to lead to protective immunity, or it can go to lead on to disease. Now, one of the issues with Mycobacterium tuberculosis is that I've pointed out at the bottom is reactivation. I'll come on to later in terms of the statistics in terms of tuberculosis as a disease worldwide. But the term reactivation, it means that someone could be very healthy and they could have Mycobacterium tuberculosis residing within their macrophages, but everything remains in check in terms of the immune system. So really, you don't actually go on to develop any clinical symptoms of the disease. What happens later in life, either through sort of issues in terms of one could be HIV infection, sort of co-infection, or in terms of transplants, where the immune system starts to wane, you then start to find the bacteria to come back, reactivate, and cause disease. And this can happen many, many decades later in life. So what are the issues with tuberculosis? It is really a disease that we associate with poverty and poor healthcare. And the factors that sort of lead to these sort of increasing rates, clearly one of the major sort of factors is HIV. And what we're finding is an increasing trend where if we look at the TB endemic population and the HIV endemic population and this sort of centre sort of cohort of clinical TB cases, what we're finding is an increasing trend this way, where we're finding a higher proportion of case rates of TB within HIV individuals. It should be also pointed out that in terms of HIV individuals, TB is the largest killer within that sort of cohort. Another problem with tuberculosis is that it's spread very easily. So this is a sort of graphical representation in terms of the number of individuals it will take to infect different individuals in different case scenarios. So at the top we have sort of one person who went on to infect 40 people in a bar. The next is an airline person who went on to infect 4. Next you have a person in a US shipyard who went to infect 400, and so on. So you can see it's a very contagious disease. So really what you're finding is that one individual can go on to infect many hundreds. So those are the sort of primary reasons in terms of why tuberculosis is increasing. One is HIV, one is ease of infection. The second really comes down to what Mark was talking about, and we had the interesting discussion about antibiotics. Now, this is the antibiotics that are used to treat tuberculosis, and these are the number of antibiotics that you take on a daily basis. So the problem comes in that you're taking 4 different drugs through what we call the Directly Observed Treatment Short Course DOTS, which was established by the World Health Organization. You have rifampicin, isoniazid, ethambutol, pyrazinamide, which are given daily for 2 months. Then you have a continuation for another 4 months of rifampicin and isoniazid. So this is where we start to run into problems. You have a multi-drug therapy which lasts for 6 months. And if anyone in this room who's had flu or the cold, who then goes on to take antibiotics, it's quite difficult to actually just finish a course of 2 weeks. And you can imagine trying to finish a course over 6 months is quite problematic. And this is where we run into problems in terms of drug resistance. One of the sort of outlying factors in terms of when we look at these drugs, if we look at rifampicin, it was discovered in 1967, isoniazid 1951, ethambutol 1965, pyrazinamide 1952. So all these drugs came about in what we refer to as the golden era of chemotherapeutics. And really there's really been no new TB drugs developed since that time. And an interesting sort of point at this point in the talk is that when we look at the mode of action of these drugs, they all target some aspect of cell wall synthesis, which we'll talk about later. There are alternatives to chemotherapy, and this is a sort of example of a guy from Russia who was infected with tuberculosis, and you have surgery, which is a last resort, as we can see. So really, in terms of tuberculosis and global trends, we have around 8 million new cases each year. There's 2 million deaths. And really, when we start to look at the epidemiology of the disease and start to map onto this world scenario in terms of multi-drug resistant tuberculosis, which we refer to as MDR-TB, and we also have a new sort of phrase that we use now, which is called extensively drug-resistant tuberculosis, XDR-TB, where we have resistance to all the frontline agents used to treat tuberculosis and resistance to some of the second-line injectable treatments that we have to tuberculosis. So what you can see is that we have really high cases in Asia, parts of Africa, and parts of South America. And if you go back to the beginning of the talk, when we talked about sort of poverty and local care in terms of health, you know, these are the sort of regions that we would anticipate to have really high sort of incidences of tuberculosis. So really, if we come back to the sort of issue in terms of reactivation and case rates, really a third of the world's population is infected with tuberculosis. But not everyone goes on to actually develop clinical symptoms of the disease. And again, it's sort of reiterating that we have 8 million new cases, 3 million deaths each year. And really, if we look at a sort of case study in terms of incidence of TB remains high in less developed countries, really there's been no change in terms of the incidence of tuberculosis in India since independence. And this mortality rate exceeds that associated with other infective organisms. So really, you know, where did we go wrong with tuberculosis? Now, if we look back at the earlier slides in terms of when the agents used to tuberculosis were discovered in the 50s and 60s, really we've really overestimated the power of existing treatments, and we've not really followed up in terms of new developments in terms of antibiotics. Another area is that we've changed as a society in terms of immigration and travel, and that has had a profound impact in terms of tuberculosis incidence. And the other major contributor was that we didn't expect the HIV epidemic. So what are we looking for? You know, going back to the discussion that we had, you know, I disagree with Mark a little bit in that, you know, we do need new anti-tuberculosis drugs. We need new drug targets. If you think of the way science was done sort of 50, 60 years ago when these drugs were discovered, they came through rather sort of simplified phenotypic screening platforms where people or pharmaceutical companies would take compound collections and screen them for inhibitors. And that would be their sort of proof of principle. They would identify agents that will kill the bacteria in the laboratory, and then these would go into clinical trials. What's happened is that as science has improved, and really those drugs were all discovered in the 50s and 60s, but no one actually knew how they actually worked. And really, we didn't discover their sort of mechanisms of action in terms of what genes they target, really until 40, 50 years later. Now, there's been a sort of change and shift that went with that in terms of saying, well, if we've got targets, why don't we just screen for new inhibitors against the targets? Now, the problem comes back in that tuberculosis has this sort of wily sort of cell envelope coat. And that is really a formidable barrier to cross. So people were trying to develop new drugs in vitro, in the laboratory, in test tubes, which make fantastic inhibitors in enzyme assays. But these can't cross that wall, so they're effectively no good as drugs. So there's been a big push now to go back to what we were actually doing 50, 60 years ago, but in a smarter way in terms of trying to do phenotypic screens again, but using modern genomics in terms of developing drug-resistant mutants to then identify their targets. Now, the benefit there is that you're starting with a lead molecule which you already know is active, and you're using modern genomics to identify its target. We also need to have a better understanding of tuberculosis pathogenesis, and rightly, we do need better vaccines. So our overall strategy at Birmingham is really to try to identify drug targets, use phenotypic screens, use functional genomics, and try to take these candidates further in terms of focused studies. Really, I've been really focusing in terms of trying to use a multidisciplinary approach. My background was originally as an organic chemist. I'm far from an organic chemist now, but you can do a lot of different things with a lot of different backgrounds in terms of training. So, taking a chemist, you can become a biochemist, you can do immunology, you can do genetics, and really you can do structural biology. So, over time, as an individual, you'll develop different techniques and apply them to the problems in hand. So I'm going to finish up on the cell wall of Mycobacterium tuberculosis. So really it has an underlying peptidoglycan. Attached to this is a structure which we refer to as arabinogalactan, which is abbreviated as AG. I'm going to show that I'm a chemist by heart, in that we have a phosphate group here, which is attached to the muramic acid of peptidoglycan. And then we have this disaccharide bridge of two sugars, which are unique, in that we have rhamnose and acetylglucosamine. And then, as arabinogalactan suggests, we have this particular polysaccharide made of two sugars of arabinose and galactose. These are fairly unique in that they're both in the furanose ring form, and they have very unique sugar linkages. Attached to the termini, we have these long-chain mycolic acids, and there's different varieties of these depending on different sort of functionalities. But one of the characteristics of these fatty acids are, when you think of a typical fatty acid, it's C16, C18, C19. These are fatty acids that are up to C90 in terms of carbon length, so very hydrophobic. And intercalated within these mycolic acids, we have a variety of unique cell envelope lipids. We have things called thioceryl dimycosarosates, abbreviated as PDIMs. We have diacyl trihaloses, phenolic glycolipids, PGL. So really, going back to what I was mentioning earlier, this really gives a very sort of hydrophobic sort of outer shell to the bacillus. And this is one of the reasons why conventional antibiotics are not effective against Mycobacterium tuberculosis. But coming back to the phenotypic screening, was that when isoniazid INH and ethambutol EMB were discovered in the 50s and 60s, really they were shown to target aspects of cell wall synthesis. Isoniazid targets aspects of mycolic acid synthesis, and ethambutol targets aspects of arabinogalactan synthesis. So really, many of these enzymes are essential that make up this cell envelope, and they are attractive drug targets in their own right, and they need to be exploited. I'll stop there. Okay.
Thank you very much, Dan. Are there any questions before we move to the next speaker? Yes. Oh.
In terms of the drug cocktail implications in tuberculosis, you said prizes in mind, the mode of action is unknown. So I was just wondering, why do you give that to patients if we're not quite sure how it works, or is it just observational evidence that it seems to specifically describe what to do?
If I understand your question correctly, really, when we start to look at resistance, and what we have to remember is that there are different levels of resistance. So you could have resistance to isoniazid, which is what we would call at its minimum inhibitory concentration. But the actual levels at which you give isoniazid to an individual are much higher in terms of serum concentrations. So there will be a spectrum of resistance that you'll see to the different antibiotics.
No, I'll just answer that one, because I think I see where you come from. In days gone by, we used to use an empirical approach. If antibiotics work, you had agents that worked. You tested them on animals and humans and they didn't kill people. They were selected as agents that you could use. Now, if you want to get a new drug on the market, you have to actually understand how it works before you will get regulatory approval to use it. Pyrazinamide actually is one of those unusual agents where the resistance doesn't actually come from target site modification of the thing that it's actually doing damage to in the cell. So it's, again, there's a little twist there. And in fact, isn't it about 2 weeks ago, 2 months ago, they actually discovered the basis of pyrazinamide action. So it has actually now been, it was in Nature a couple of months ago that finally has been nailed down. But you're right, it's interesting to contemplate the fact that actually much of medicine in the past was empirical. It's things that work. Now we have moved forward with kind of ratchet effect in what's required. You have to understand why things work before people will let you use them. In fact, it's interesting because you're going to hear later in drug discovery that 2 decades ago, everyone would say you have to understand every enzyme, active site, and everything perfect, design the drugs perfectly. And people are now realizing, well, actually, that may not be the only way forward. Perhaps it's just good to screen lots and lots of compounds empirically and see what works. And then use your smart intuition to actually tweak things to make them work even better. But it's a mixed, we're using a mixed approach nowadays.
It's probably worth saying that, you're all going to see TV over the walk of medicine you go into it. And there's an old adage as well, that it's also the great masquerade. So, you will be faced in your lives with people whose clinical features don't quite fit something. They look sick, and the message is always TV. There used to be an adage, you would never let somebody go to their grave without a course of antituberculous chemotherapy if you were so uncertain of what was going on. So it is a great masquerade. The other thing I was just going to ask the panel, of course, is these drugs, like all drugs, are not without side effects. And another flip side of not having a single effective agent is you end up giving 4 or 5 drugs that cause neuropathy up to problems. That's another issue.
There's a lot wrong with the current drugs, it's true. And the other point is that we have to give combinations of drugs because the bacterium does become resistant if you give a single agent. If you give a single agent, there's about a 50% chance the patient will relapse with drug-resistant disease. So it's a very wily organism. Anyway, I'm going to switch now back to I'm going to talk about E. coli for a few minutes. It's a very versatile bacterium. It was actually discovered in 1885 and then renamed E. coli. Sorry, I forgot to put italics on my species name there. In 1919. This is Theodor Escherich, after whom E. coli is named. And in fact, just as an aside, we've got the original E. coli that he isolated here in Birmingham, and we're genome sequencing it at the moment, and we're interesting to find out what that's going to be like. In a sense, E. coli has become domesticated. There was this particular strain, E. coli K12, isolated from the feces, I should say feces, not faces, feces of a convalescent diphtheria patient at Stanford in 1922. Nothing special about the patient, nothing special about the E. coli. But that particular strain then got adopted by the scientific community, just as a kind of historical accident. These guys, you may have heard of them, Beadle and Tatum, worked on Neurospora and came up with this one gene, one enzyme hypothesis. And then one of them, Tatum, turned his hand to E. coli, started working on K12, irradiating it, seeing if he could create mutants. And then his student, Lederberg, in 1946, actually showed that E. coli could exchange DNA. Different strains could exchange DNA through a process known as conjugation, where plasmids pass from one strain to another. It's the bacterial equivalent of sex, and Lederberg got a Nobel Prize for that. E. coli actually has featured prominently in the history of science and the history of molecular biology. This paper here from Lurie and Delbruck, they showed there that mutations occur before selection. So this was one of the things that Darwin had kind of had left hanging as to his idea was all the variation was there and natural selection selects from what's already there. It doesn't actually change that variation. And they actually showed that by looking at the effects of bacteriophage on E. coli and showing that resistance to the bacteriophage was pre-existent in the population. They did some quite sophisticated statistics to show that. In fact, DNA being the genetic substance was first shown in E. coli, or the interaction between E. coli and the bacterial virus that infects it, the bacteriophage. That was first nailed down there by Hershey and Chase. And there's the famous Mazes and Stahl experiment where they showed the semi-conservative replication of DNA using E. coli. They showed that basically the two strands separate and then one of the old strands is there, a new strand is made next to it. So E. coli really has been at the centre of science over the last 80 years or so, 90 years. In the 1950s, Jacob and Monod actually started studying the genetics of E. coli, working at how genes were regulated. And they came up with this idea of the operon. There was a string of genes in a row, all regulated by the same regulatory signals running off a single promoter. And in fact, Monod actually came out with this famous phrase, kind of Delphic utterance, that all that is true of E. coli is also true of the elephant, basically hinting at the fact that really E. coli is the model organism, biology's model organism, premier model organism. And we know more about E. coli than we know about any other organism on the planet. The genetic code, that was all sorted out in E. coli in the 1960s. And in the 1970s, E. coli was adopted, particularly by Boyer and Cohen in Stanford, as the basis of genetic engineering. And nowadays, millions and millions of diabetics around the world are relying on insulin that's actually produced in E. coli. We don't use pigs anymore. It's all genetically made in E. coli. So E. coli has become the kind of workhorse for biotechnology. If you're interested in the history of E. coli as a model organism, there's a book from a friend of mine here, Carl Zimmer, called Microbes, where he actually traces all these little stories and gives you the vignettes of the people working on E. coli over the years. In Birmingham, lots of people work on E. coli. I just went to PubMed and had a look, typed in the postcode of the university and E. coli, Escherichia coli, and just in 2 years, over 50 publications coming out of this place. I was surprised quite how much is going on. Much of it I knew about, but there are a few things I didn't, and I can't cover any of it, all of this in the time available. Just a small part of it. I can just give you some vignettes from what's going on this campus in regards to E. coli. This is a colleague of mine, Steve Busby. He has continued that thrust of using E. coli as a model organism to understand basic biological questions. He has been studying how you get transcription started, the regulation of bacterial transcription initiation. How do you actually start transcribing a gene, and how does the RNA polymerase dock with the promoter and actually get going? And he's written a review recently on this. He has been so influential, his work has been so well recognised in this, that just a couple of years ago he was given a fellowship of the Royal Society, FRS. So that's about the highest honour you can get apart from a Nobel Prize in science. And as a Republican, I think the Royal Society is about the only society that has the word royal in it that I would like to join. I don't think it's going to happen, but Steve Busby is one of our FRSs. There's another colleague of mine, Ian Henderson. I just shamelessly stole that from his Facebook page and then asked his permission to show this silly picture of him on holiday with a fish. But it epitomises the guy, so he is an outgoing guy, I'm sure he won't mind. He has been studying the cell envelope in E. coli. So E. coli has a cell envelope. It's not quite as sophisticated as that of TB, but in some ways it's got an added twist because it's got an outer membrane. It's got two membranes, an inner membrane and an outer membrane. And quite how you get proteins into that outer membrane, which is the part that's actually facing the outside world, is an interesting conundrum. And Ian has been working on a complex known as the BAM complex, actually involved in sorting proteins into that outer membrane and getting them to fold upright and be in the right place. And he's one of the world leaders in this, working on this here in Birmingham. I mentioned about E. coli as a model organism. Well, a few years ago, I actually started getting a bit cross because there are people who work on it as a model organism that actually really do have tunnel vision. They think the whole of E. coli is E. coli K12. And so, with a couple of colleagues, we actually wrote a little piece saying that actually you've got to be a bit more savvy about this and actually look on a broader view of E. coli. And also, if we look at the K12 lineage, it's actually a very atypical E. coli because it's been kind of battered around in the lab. It's become really quite different from E. coli's out there in the wild. And people have started to look on it as kind of like a platonic ideal of E. coli, it's like an icon of E. coli and of biology, rather than realising that actually biology is messy in all its variation. Let me just give you perhaps a rather strained analogy here. You go from this fresh-faced thing out there in the wild, this is your E. coli in the real world. Once it gets into the lab, it starts to become a bit disgraceful and kind of, you know, not behaving quite as it should. And before you know it, it's actually become something. It's become an icon, something really totally devolved from the real world. And this has been you know, we've had some interesting discussions about this. It's an interesting discussion about model organisms in general. We have to use model organisms. We have to use inbred mice. We have to use particular strains of inbred mice. But we have to be very careful about not over-interpreting our data. You know, what works in the mouse generally works in humans, but not always. And what's true of mice is not always true of humans. In fact, it's epitomised by a phrase that Alfred White said back at the beginning of the 20th century. He said that all scientists should seek simplicity and then distrust it. And I think that's the view that we're getting out of E. coli here when we study E. coli K12. In fact, the idea that E. coli causes disease and is diverse goes back a long way. It's first recognised over 100 years ago in this paper here that E. coli actually caused urinary tract infection. Over the 20th century, it became clearer and clearer that E. coli was actually a pathogen and a very diverse pathogen. So in 1944, we got a serotyping scheme. We got 1945, the idea that E. coli was actually causing diarrhea. We then got in the 1970s recognition of different kinds of E. coli causing different kinds of diarrhea. Recognition that some strains can cause meningitis. And even in as late as the 1980s, we started recognising new kinds of E. coli causing diarrhea. So it really is a very versatile pathogen. It causes disease in many kinds of animals, let alone humans. And in humans, it can infect the gut. Of course, it's the predominant cause of urinary tract infection. You can get into the bloodstream infection, you get meningitis. And among the infections in the gut, we have this profusion of different kinds of E. coli that have been named. Enteropathogenic, enterotoxigenic, enteroinvasive, enterohemorrhagic, enteroaggrative, diffusely adherent varieties. And we now recognise that Shigella, although it has this genus name, is actually just a variety of E. coli. So it's a tremendously versatile pathogen. Coming back to Darwin in the drugstore, E. coli, like many organisms, has over the recent years seen this dramatic rise in resistance. Here's another of my colleagues, Peter Hawkey, has been charting the spread of one particular enzyme, a so-called extended spectrum beta-lactamase. So it basically chews up penicillin and its relatives, but actually chews up not just the early penicillins, but even the later derivatives and the later derivatives of cephalosporins that we use, which we think of as being very sophisticated agents that we've derived in the last few decades. They are now being chewed up by these extended spectrum beta-lactamases, and there's one particular one, the CTXM family of these extended spectrum beta-lactamases that are being found in more and more settings around the world, around England, but also in China, in India, and elsewhere. As you can see, it says here, in the UK, first occurred in Shropshire. Think of rural Shropshire as a sort of place nothing much happened. That was the focus for the emergence of this resistance in England. We've been charting the genomic diversity in E. coli since the first genome sequences were done over 10 years ago now. And this, we actually run in my group a little database called XBase. and this is a cartoon produced from it. In the middle there is K12, and we've coloured its genome according to the nucleotide composition, the GC content, but the ones around it we've coloured in green if it's the same as K12 and in red if it's different. And it's clear that E. coli genomes are like a mosaic. There's a core part of the genome which is shared with K12 and with others, but there are lots and lots of things that are variable popping in and out of that genome all the time. In fact, we summarised this in a paper in a review in Nature a couple of years ago, looking really just at the various forces that act on bacterial genomes, and horizontal gene transfer is one of the major forces. So what this means is instead of a cell dividing into two and passing its genes on just to those two progeny cells, you can get the movement of genes horizontally from one cell to a totally unrelated cell. And this happens on mobile genetic elements, bacteriophage, bacterial viruses, and plasmids very commonly. In terms of genome sequencing, here's just a few screen dumps to show that we've been quite active in the last couple of years, sequencing various kinds of E. coli here in Birmingham. Ian and myself have been doing that. Ian's actually taken the effort of writing them, all these things up recently, and we can see we've got all sorts of things here. We've got an enterotoxigenic E. coli, we've got the prototypical enteraggregative E. coli, we've got an E. coli that's come out of a patient with Crohn's disease. One interesting paper from the recent years is this one here, where we actually looked at a variety of E. coli called enterotoxigenic E. coli. And so this E. coli causes disease by producing some toxins, enterotoxins. The toxins are actually encoded on a plasmid. It's a circular piece of DNA that replicates independently of the chromosome and actually can move from one bacterial cell to another. Now the question before we did this study was, actually, is there such a thing as enterotoxigenic E. coli? Like there's a lineage of E. coli that carries that plasmid, but actually it's quite a tightly defined lineage. The chromosome is actually adapted to the plasmid. They go very well together and so forth. Or is it that the plasmid actually is promiscuous and can go into any old E. coli and turn it into an enterotoxigenic E. coli? Well, when we actually did this survey here, we looked at the genetics of the genetic survey of lots of different enterotoxigenic E. coli, and what we found was that actually the plasmid will go into any old E. coli lineage. Many different lineages have had this plasmid go into them. And so it's the plasmid that is actually, if you like, at the heart of the disease. And the chromosomal background, the lineage that you're going into, is relatively unimportant. I had a similar paper a few years ago in PNAS where we looked at another kind of E. coli. This is E. coli 0157. It's a kind of E. coli that we often call enterohemorrhagic E. coli. And one of the virulence determinants of this E. coli is its ability to produce what's known as a type 3 secretion system, which is a kind of molecular syringe that injects virulence factors into cells. And we did a survey. We thought, well, there were probably more effectors for these virulence effectors than had previously been suspected. And so we did a survey that included bioinformatics and experimental work. And we showed indeed there were a very large number of these effectors, but they were scattered throughout the chromosome. And when we looked carefully as to where they were sitting in the chromosome, we realized that they're actually sitting in what we call prophages. So these are bacterial viruses, bacteriophages, that have actually integrated themselves into the chromosome. And this is part of the lifestyle of many bacteriophages, that they can live as viruses out there in the outside world, and they can infect cells and just destroy the cell, turn it into a bacteriophage factory. But they also have the choice of just hunkering down, sticking themselves into the chromosome and living as part of the cell for many generations. And this figure here, the green arrows there are these virulence effectors, and they're all shoved into the one end of these bacteriophage genomes in the genome, and they have this characteristic of having an extreme GC content and being located at that particular point. So in a sense here we've got the phages also carrying virulence factors, just like the plasmids did in the previous study. So actually we're seeing in E. coli a crucial role of mobile elements, enterotoxins on plasmids, type 3 secretion linked to these phage. the effectors on those phage actually not just being in the chromosome, but out in the outside world, in sewage and so forth. There are huge numbers of these phages, and so we sometimes use the word metagenome to talk about all those phage genomes out there. And there's huge amounts of recombination going on and exchange of DNA and genes in those phages. And so one of the former fellows who used to work with me actually said, well, if you think about it, maybe E. coli is just a kind of vehicle. And the way we should think about infection is that actually these infections are just plasmids messing with us, or they're phages messing with us, and the E. coli is just carrying those determinants as a sort of, it's a bit like, you know, Richard Dawkins' selfish gene idea that we're all just bags of genes, you know. In a sense, E. coli is just a bag of, a platform for the launch of plasmids and phage to attack humans. So that's me finished for this part, and I'm going to pass over. If there are any questions quickly at this moment.
Mark, in view of time, I just wondered, because actually I know what you're going to say vaguely about Germany, which would be really useful maybe, just to briefly, if you can cut that back to maybe 5 or 10 minutes, and we'll maybe finish on the new drugs. Is that okay?
So you want me to go straight to Germany now?
I think that would be good, because it's extremely relevant actually.
Right, okay, so fast forward to Germany, and I'll try and speak as quickly as I can, because we are slightly running behind time. Okay, so what I'm going to talk about now is our experiences with E. coli over the summer, this German outbreak that you may have heard of. And this German E. coli actually causes a syndrome called hemolytic uremic syndrome, which is characterized by, it's produced by types of E. coli that produce a sugar toxin. It's characterized by bloody diarrhea, damage to the kidneys and brain, you get anemia, you get loss of platelets. This is the sugar toxin. It's a toxin that has an active subunit, an A subunit on the left there, and then it's got these 5 B subunits, or binding subunits, that allow it to bind to the surface of cells. And this is at the heart of hemolytic uremic syndrome. This is what causes that syndrome. I give this talk to the general public, so this is, perhaps I'm talking down to explain what sequencing of genome is, but it's basically turning that material thing, DNA, into something digital, into a DNA sequence. It's a bit like taking a scanner and scanning a book and then turning it into something digital in a sense. So when we sequence the genome, we're capturing the instructions for making the cell. Just as a quick aside also, you might have heard Thomas Edison's statement, genius is 1% inspiration, 99% perspiration. But actually, much of the advances and staying at the cutting edge in laboratory science is actually now due to instrumentation rather than inspiration or perspiration. Another interesting comment there is actually from Faraday. Typo there, it's not David, it's Faraday, said work, finish, publish should be your slogan if you're a scientist. But the way science works is that actually most people don't tell anyone about what they're doing until the work is published. And it's all kept secret, and many times people are in competition and they keep everything quiet. And we're going to just challenge that now. So here in Birmingham we do genome sequencing. A colleague of mine, well, bioinformatician who works with me, Nick Lohman and myself, we run a blog, and this is Nick's blog entry from August 2009, and you can see what a geek he is. He says, I was anticipating today like a kid waiting for Santa Claus. What could provide such excitement? Nothing less than a brand new 454 DNA sequencer. So this is a new kind of DNA sequencer, genome sequencer, that works very much more effectively and quickly than anything that had gone before. So we were very lucky to get this back in 2009. Here's a colleague of mine, Charles Penn, looking a bit of a lemon there, standing next to the crate as it arrives. Nick took several photographs of it as it arrived, so geeky was he. Fast forward though, I get a bit geeky as well and blog and stuff, and earlier this year I actually won a new kind of instrument that has just come on the market called an ion torrent personal genome machine. So I filled in a form towards the end of last year. They wanted to bring this instrument into Europe, and they said they were going to give away 2 as a marketing exercise. So I filled in the form, took me 20 minutes, and I won. Out of 125 entries, I was one of the 2 that won. Someone pointed out that I shouldn't just say it was 20 minutes. It took me 20 years to get to the stage of being able to write a winning entry in 20 minutes, I have to say. But so we won that. We won that, partly on the back of the fact we already had a 454. And so we were poised and ready to go. We got this instrument, and we were just gearing up to thinking, how are we going to start analysing data from this instrument? Each instrument has slightly different ways of creating data, and slightly different things come out of it. Ion torrent is actually one of the new generation of what we call benchtop sequencers. So it's about the size of a laser printer. You can see it looks kind of funky. It's got this PlayStation symbols on there. It's actually got a dock for an iPod or for an iPhone in there. When I first saw that, I thought, well, maybe you could control the instrument through an iPod. But in fact, it's just a gimmick. And you can decide whether you think that's cool or just cheesy. I don't know. It's what it does is it has this chip, which is, in fact, effectively a massively parallel pH meter. And what it does is that you've got millions of wells, each one with a sequencing reaction going on. And when you get incorporation of a base during the sequencing reaction, you get release of a proton. And it's the proton that's actually detected by the instrument. It's got a 3-hour runtime. And the remarkable thing is it only costs a few £100 per run. So we can start sequencing genomes for cost of hundreds of pounds, or even less, maybe tens of pounds, which is remarkable. The TB genome, I think, was several £100,000 when it was first done. Similar with the E. coli genome a decade ago. The prices have dropped dramatically. So quickly looking at E. coli in Germany, Germany was struck by this E. coli 0104H4, a rather unusual variety of E. coli. It caused over 4,000 cases, over 40 deaths. The epidemiologists actually linked it to sprouting seeds, the fenugreek. It took them a while. They had, at one stage, issues with cucumbers. In fact, speaking to the guys in Hamburg, they said that they had a lot of gay men coming in with cucumbers, saying that they'd been doing things with these cucumbers, and did that place them at risk. But in fact, it wasn't the cucumbers, it was these sprouting seeds. We have the high risk of hemolytic uremic syndrome in this outbreak, much higher than usual, and for some reason females seem to be more at risk so about 55% of the cases were female in terms of total cases, 65% of those with hemolytic uremic syndrome. I don't think they've got to the bottom of that yet. One potential explanation, which I've thrown out, is that perhaps real men don't eat bean sprouts, and it might be that's just that, the men don't eat the salads, they go and eat the steak. That might be an explanation. You can see that the outbreak was concentrated in northern Germany, but it actually spread throughout the whole of Germany, and several other countries were involved. And here's a typical kind of epidemic curve there, at its peak around the 3rd week in May, and then slowly petered out into June, and there were a few cases just after that. So the centre of the outbreak is a collaborator of ours, Herr Dr. Holger Roeder at UKE in Hamburg. He told me they just had all the wards full up with these patients. They had to open up new temporary accommodation. It was right in the middle of the storm. And he was thinking, well, what could he do scientifically, kind of interesting, with the material he had, all these strains and so forth. But he didn't know what to do. He was actually just a humble microbiologist. He wasn't a molecular biologist. Well, he was kind of molecular, but he certainly wasn't a genome sequencer. So what do you do in such circumstances? Well, I don't know, this audience may be too young to remember what you're supposed to do in such circumstances, but when I was a boy, this is what you did. You called International Rescue, and International Rescue came to your assistance. I hope you're familiar with what happens next. Basically, you call International Rescue, 3, you get Thunderbirds come and help you. 1. In fact, it wasn't quite that dramatic. But he did get help from the international community in a big way, in a totally unexpected way. So one of his Chinese associates actually said, why don't you send the isolate to China, to this new big genome centre called BGI Shenzhen. And so that's what they did. They didn't send it in Thunderbird 2, they sent it by conventional air freight, but they actually sent the DNA to BGI Shenzhen. Now BGI, just like us, had just taken receipt of one of these ion torrent instruments. And so they thought, well, let's just see, let's have a play, let's sequence it on the ion torrent. Now, what they also did, though, was something quite inspired. They released the DNA sequence, the genome sequence, from their run into the public domain immediately. As I say, normally people keep everything secret until they're ready to publish, but they said, oh no, let's just put it out into the public domain. And this guy here, Nick Lohman, bioinformatician who works for me, was actually just noticed this. He was actually at a conference on bioinformatics of healthcare, and he said, look, they've released 5 runs of ion torrent data. Not clear what the restrictions are, but let's just get on. Shall we just crowdsource some analysis? So he was just saying to all the other bioinformaticians he was shooting the breeze with at the time, very heavily into Twitter as well. Because our blog had a lot of followers on Twitter and following the blog. He said, look, let's just have a go at this. And so what he did was he assembled the data. So basically, when you have a genome, it starts off as lots and lots of pieces of a jigsaw puzzle. You just have lots and lots of short sequences. You try and assemble them together, try and make sense of the genome. So he did this first pass assembly. He was primed to do that because he'd been playing with ion torrent data because we had our own ion torrent. And he released his assembly straight back into the public domain within a few hours of the BGI data going up. And then something remarkable happened. Within 24 hours, at least we said, the genome was assembled. Within 2 days, someone else had assigned it to an existing lineage. We had strain-specific tests within 5 days. Within a week, over 24 reports had been filed on the biology and evolution of this strain on an open source website. This guy here, who blogs under the name of Mike the Mad Biologist, his real name is Mike Feldgarten, he came in with a few hours of Nick putting up the assembly, and he looked, using an older typing technique known as multi-locker sequence typing, he looked to see if this strain was actually as new as people were saying. The Chinese had said, oh, it's a new monster hybrid strain, never seen before in the world. He said, well, actually, no, there was something seen in Germany about 10 years previously. That kind of gave some context to things. But then things really did go international. I want to say about international rescue. You know, we had people from 4 continents actually contributing to the analysis of this strain. Those 3 I mentioned put up there particularly contributed. But there were people working all over. You know, Kat Holt was in Australia, Dave Studholme in Exeter, Marina Marinkis in Spain, but Mike Feldgarten in the US. But there were groups also at BGI and in Hong Kong working on this. It really was an international collaboration. And this just shows you a screen dump from that open source wiki, the first week's worth of analysis, or a couple of weeks' worth of analysis. And you can just see this tremendous load of people actually lodging data, lodging their analyses on there, communicating via by blogs, via this wiki, via Twitter. And you probably can't quite read it, but just looking at the names, you can see that there's people with Polish names, there are people with Spanish names, there are people with Muslim names. This really was British names. There's international collaboration. It's actually caught the eye of the scientific press. Science actually ran a paper article on this. Nature, on their blog, mentioned this. It caught the attention because it was rather unusual to see so much analysis going on so quickly. So what were the takeaway messages from all this? Well, the clear point is that these pathogens don't bother with passports. This is not a new strain, something similar we've seen in Germany, but also in Korea. And the closest genome sequence strain was actually isolated from the Central African Republic back in the late 1990s. Actually, this STEC in Germany comes from a lineage known as enteragrative E. coli, which normally circulates in human populations. Normally, when you have sugar toxin producing E. coli and you have hemolytic uremic syndrome, things like E. coli 157, they actually come from cow feces. You can usually go and find somewhere in which there's been contamination, either the meat has got contaminated in the abattoir or vegetables have got contaminated out in the field or somewhere with animal feces. But this was not like that. This is a kind of E. coli that circulates in the human population. It's clear that the bacteria evolved very quickly, jumping around these virulence factors. And also, worryingly, antibiotic resistance was seen in this E. coli, even though nobody was using antibiotics to treat diarrhea. We usually say don't treat uncomplicated diarrhea with antibiotics. And if someone has hemolytic uremic syndrome, we definitely say don't use antibiotics because there's fairly good evidence that it makes it worse. So even though people weren't using antibiotics, there was antibiotic resistance in that strain. I think it's also important to stress that infection, although we kind of say, oh, it's marginalised now, it's not a problem for us in the West, this was one of the most advanced societies on the planet, and it was decimated by infection for a few weeks in the middle of the year. What we did, this kind of effort, we actually gave it the name open source genomics, where we see that there was this high throughput genomics, sequencing things very quickly, releasing data very quickly, this very liberal approach to data release, and then the so-called crowdsourced analyses. So by crowdsourcing, we mean that this is not planned. You're not saying we're going to go to the public health authorities in England and ask the person that is the bioinformatician there to do this. We just said, anyone in the world who wants to help, come and help. And they did. It's clear that social media, blogging and Twitter can augment the usual channels of academic discourse. There are many scientists who still think that blogging and Twitter and all this is just sort of a waste of time. In fact, in that film, Contagion, there's there's a lot of good one-liners in that film. One liner was that blogging is just graffiti with punctuation. I don't share that view. I think that blogging does have a place, and so does Twitter. Have we really broken the mould? Well, it may be that this kind of approach is appropriate for public health emergencies. Whether it will work for ordinary science is unclear. I think I'm going to have to move a bit quicker. So the clear point is that we now can sequence things quickly enough to actually have an impact on the real world. Lady mentioned earlier about these bacteria mutating all over the place and spreading and being, basically bacteria engaging in global group sets, sharing genes across the planet. Can we beat them? Well, this is, it's the wisdom of the crowd. Nowadays, with the internet and these technologies, the whole planet can focus its intellect down onto these problems. There's a couple of taglines that we used. Knowledge is power. The Germans wanted, misspelled there, but Vorsprung-Technik, to illustrate the point. One of the things that we were very pleased to do was we actually wrestled down all this stuff going out into the social media and so forth, into a publication. It was a little bit of a rocky ride, but we'd actually managed to get this distilled down into a publication, and we were very lucky that we actually got it into a very high-impact journal, New England Journal of Medicine as well, so that was, we felt very pleased, and you can see us here having some champagne with our German collaborators at the top there. And here, this is Dave Rasko, who actually had a paper back-to-back with ours in the New England Journal of Medicine. He came to Birmingham a few weeks ago, and we had a nice drink with him as well. I'd like to finish with a quote, actually, that relates to Birmingham. Some of you may be aware that back in the late 18th century, there was a group of intellectuals, entrepreneurs, medics, who met together, known as the Lunar Society of Birmingham. They included Joseph Priestley, included Erasmus Darwin, Josiah Wedgwood, and they met not far from here, up near Handsworth. And this is a quote that Joseph Priestley said about the Lunar Society of Birmingham. And I think the same quote, the same internationalist kind of viewpoint is actually epitomised by the study that we did over the summer. And that's me finished.
Good, that's terrific, Mark. Thanks very much. I'm just conscious of time, and I know we've got two fantastic final speakers. So, do you think we should just move on?
Yeah, I think
just in 5 or 10 minutes each, just get an update from
our
speakers, and then we can leave 10-15 minutes for discussion.
Yeah. Okay. Sorry.
Thank you. We need to get people mic'd up.
It's worthwhile saying, while you're talking, of course, that William Withering was the first one of the founder members.
Yeah, exactly. William Withering as well. And there were lively arguments between William Withering and Erasmus Darwin about who got to foxglove first and all that. Yeah.
And William Withering's, if you go to Edgbaston Clubhouse, that was William Withering's house.
Is Luke on this? Is Luke? Okay. Can you start?
Okay, we'll have the last two talks and should leave us with time
discussion.
Right, so I'm going to pick up from where Del left. What I'll do is I'll try to give you a sort of brief glimpse into what goes on in the lab. So all of you are going to be at some stage into your clinical training. You're going to hand out prescriptions. It was going to be quite likely that these are going to be antibiotics. What I'd like to sort of bring out in this talk today is that that prescription that you're handing out, perhaps if not years or decades, that particular drug that you prescribe to a patient must have gone through a clinical trial. And probably decades before that, somebody in a lonely lab somewhere must have found out that this drug acts against a particular enzyme, against a pathogen or a cancerous cell. And so what I'm trying to say is I'm going to talk a little bit of one end of the spectrum is how do we go around making these discoveries? How do we go around finding out more about pathogens, or for that matter, any other disease that afflicts humans? What I'll do is I'm going to pick, I was originally going to pick about 3 examples, but I think I'll cut it down to 2 since we've short of time. Right. So this is a sort of general scheme that I've put up about what we do in the lab, or for that matter, in a bacteriology lab that's makes use of genetics to understand more about bacterial pathogens. Geneticists are destructive in nature. I'll tell you why I say that. What we try to do is, if we want to understand the function of a particular gene, if we want to understand a particular process in a bacterium, we like to break things. We like to take away that gene and then see what happens. So it looks like a very destructive approach, but it actually ends up, giving you a lot of answers. The way we do that is the use of genetic tools. The genome sequences that are now available makes things a lot more easier for us. For example, we can use bioinformatics. So if we're studying pathogen A and we want to find out what are the virulence factors, what are the genes that are helping pathogen A cause disease, then we could get an example from some other pathogen where say a particular gene exists and that's been shown to play a role in the disease, and we find the same gene in the pathogen we're interested in. So we go ahead and knock out that gene, so make a mutant that's missing that gene. We can also have a sort of random approach. One such approach is the use of transposons. So transposons are mobile DNA that jump, in this case, randomly into different genes in a genome. So you will have a pool of mutants. You don't know each individual mutant, but you could have a screen. So that screen would then yield you a mutant that you were interested in looking at. So putting it very shortly, it's finding a needle in a haystack, but your screen would be a very strong magnet. So you take your magnet around the haystack and pull out the needle. Of course, once you've isolated a mutant, you've characterized it in the lab, you also want to know, you want to assess the effect that mutation has in an animal model or in a cell culture model. And for tuberculosis, what we end up using is the macrophage. So this is the first cell that the TB bacterium encounters. This sort of infection experiments with macrophages, although not in its whole, will not give you a proper answer, but it does tell you about the initial stages of infection. So it does contribute to us understanding a lot more of the disease. But of course, what's happening in a tissue culture plate is not the same as what's happening in a living organism. So we have to take that forward by doing studies, for example, in the mouse model. A guinea pig model also exists for studying TB. So once we get all this information out from animal infections and from macrophage infections, what that does is it tells us, one, more about the pathogenesis of the bacterium. Secondly, it tells us a lot more about ourselves. The way, of course, all these studies are done in the mouse, we get to know about how the immune system looks at the bug, and then we can extrapolate that to humans. And these studies altogether then will help us, for example, to develop better vaccine candidates for combating TB. Now, I did mention the generation of mutant strains. However, in certain cases, you cannot generate mutant strains simply for the fact that certain genes are extremely essential for housekeeping functions. So if you try to delete those genes in a bacterium, the bacterium can't survive. You can physically replace that DNA, remove that DNA, but the bacteria won't divide, and then you will not have an active culture. And of course, such genes are ideal drug targets because this is where you want to hit the bacterium when you're designing drugs. So we do have a system in place where we can study such essential genes. So there are a couple of hurdles when it comes to studying Mycobacterium tuberculosis. So it's not really an easy bug to work with. When I was doing my postdoctoral training, my supervisor, it was one of the first meetings that I had with him, and he said, "Oh, you've chosen the wrong bug to work with." You know, when I started off, I was fine, and I ended up with a lot of gray hair after about 2 years. So I didn't get any gray hair, but I can assure you that there was a lot more hair on my head 5 years ago. So it's a very slow bug. It takes about 4 weeks, nearly 3 to 4 weeks to form a colony on a plate. So one of Mark's slides showed a colony of Mycobacterium tuberculosis. Just to put this in perspective, you're working with E. coli, you plate out a culture, you get a colony the next day. So if you're a PhD student working on Mycobacterium tuberculosis and you've got 3 years or 4 years to finish off your PhD, that's a long time to wait for a result. Secondly, it also slows down the worker. This is an infection that's spread by aerosols, and it's a human disease. So it has to be done. All the manipulations that you do have to be done in a biosafety level containment facility, which means that a process that would take you maybe an hour might end up taking 2 or 3 hours in this particular environment. So what we do is, instead, we look at cousins of Mycobacterium tuberculosis. One such related mycobacteria is Mycobacterium smegmatis, that's on the top left-hand corner. This is a non-pathogenic bacterium, so you can use it. You don't need a biosafety level 3 containment for it. However, it's much faster. You get colonies in 3 days. We also use what is a surrogate, Corynebacterium glutamicum. This is again a relative of Corynebacterium diphtheria, so it's a causative agent of diphtheria. Here, the cell wall is quite similar to Mycobacterium tuberculosis or other mycobacteria, and Corynebacteria are sort of more resilient when you make mutations that affect the cell wall, so they're far easy to study in the laboratory. However, at the end of the day, we also want to study infection, and how do we get around studying infection if we have 2 non-pathogenic bacteria? So what we do is we use Mycobacterium marinum. It's a pathogen of fish, and it also is used as a model system to study infection in zebrafish that you see down there. Zebrafish has been a model for a number of diseases, and tuberculosis is one of them. And it may not look like it, but studying granulomas, that's a classical pathological manifestation of tuberculosis. Studying granulomas in fish is quite similar to studying them in humans. So what I'll do is, in the next few slides, give you 2 examples of how that sort of approach we take to finding out more about Mycobacterium tuberculosis, both as a bacterium and as a pathogen. And Del mentioned the mycobacterial cell wall, and what I'm going to focus on are those thick dark lines that you see. They're repeated along the cartoon here. Those are called mycolic acids. Very simply, they are very long fatty acids, but they're characteristic of mycobacteria. The reason we study them is right up here on the slide. So that's a mycobacterium. That's a single mycobacterium. It's been treated with isoniazid. So isoniazid is a hallmark anti-TB drug. What you can see is as time progresses from left to right, the cells start developing a lot of blebs, and eventually they burst open and die. So that's known as death by lysis. Of course, as a clinician, that's good. You're getting rid of the infecting pathogen. If you were to ask an enzymologist, they probably look at it from a different angle, thinking, oh, that's a beautiful case of one particular component, one particular enzyme being inhibited, in this case by an antibiotic. But as you can see, it's an important class of fatty acids that mycobacterium makes, because if you stop making mycolic acids, this is what happens. So isoniazid actually targets one of the enzymes that are involved in making mycolic acids. But you can see that after isoniazid, there's not really been actually no drugs there which are in clinical use that may be affecting mycolic acid biosynthesis. And there are 30 plus and still counting components that are involved in making these long chain fatty acids. So what we did was we tried to look at, well, first we tried to identify what are the other components that are involved in making mycolic acids. And the problem is, as I said, mycolic acids are essential. So if you target genes that are essential, you're not going to end up getting mutants. So we tweaked the system a little bit and generated this genetic tool which allows us to study essential genes. So what we do is, our gene delivery system is a bacteriophage, it's a virus that infects mycobacteria. We first introduce a second copy of the gene into the chromosome of the bacterium. So you can see that over here is the second copy here. However, the second copy is under a genetic switch, so we can turn it on and off using a chemical inducer. We go ahead and then knock off the original copy, which is this one. So the native copy is lost. You can keep the cells alive if you add the inducer, which is in this case, the gene is switched on. But if you don't add the inducer, then you don't get the second copy being turned on, and the cells die. And you can see here, this is an example of an enzymatic component that we had identified earlier. This again is an enzyme. It's not important what it does, but it's involved in mycolic acid biosynthesis. You can see that if you have the chemical inducer on, the cells are happy and they grow. That's a broth tube at the side of the curve, and you can see that they're happy and they grow up to saturation. However, if you grow them up in the absence of any chemical inducer, as you see in this curve, they start lysing and they die. What this allows us to do is, first of all, it lets us identify and validate that a particular gene is essential. And when I say essential, I mean essential in laboratory conditions. And secondly, we can, by using this switch and a chemical inducer that switches it on and off, we can actually study what's happening inside the cell, which we couldn't have been able to do if we had just tried to target the gene on its own, because we would have never got a live mutant with this particular gene. I'll skip this part and come to the last part, which is, again, probably up to now I've given you the impression that this particular class of fatty acids is really important for mycobacteria. However, we can target this pathway, but if you look at it from the bug's perspective, it's also very important for virulence. And I'll show you an example of one such mutant. So this again was a gene that was related to the first one that I showed you. The first one that I showed you was essential. However, in this case, we didn't have to do all this conditional switching. We just went ahead and made a mutant, and we did get a viable colony. So this gene wasn't essential under laboratory conditions. But as you can see here, this is what a colony of TB looks like. And this is what the mutant looked like. See, it's a lot smaller. And perhaps you might have already covered this in your curriculum. Two things that come to mind when you're talking about a diagnosis of TB is acid-fast staining or acid-fast bacilli. And secondly, a cording type of growth. So cording is a typical serpentine growth that you see with tubercle bacilli when they're growing in either liquid or agar cultures. And acid-fast staining is something that's hallmarks been used for ages now, and usually used for detecting TB bacilli in sputum. This is just a modification that uses fluorescence. So you can see here, the mutant that's in the middle panel has lost the serpentine growth that you see on the left. And it's also lost the ability to stain acid-fast, because what you see right below the cording panel is acid-fast staining of the same cells. So it's quite interesting because it suddenly changed character so fast once it lost the genes. And we did a biochemical analysis and found out that it still made these fatty acids called mycolic acids, but it was making them a lot shorter. So what does this mean in terms of virulence? Does it really have any relevance to the ability of the bug to cause disease? And we're quite surprised when we put this mutant into a mouse and found out that there were no signs of disease at all. So you see the panel on the left there shows the lungs of infected mice. WT means wild type, and you can see there that there's classic lesions that are being formed in the lungs, like this one over here, for example. If you see the mutant infected mice, no signs of those. And the last row over there is a complementary strain. What that means is we've taken the mutant and we put the gene back in to show that it reverts back to being a virulent bug. And on the right-hand side, you can see pathology samples of the lungs. As you can see, it's quite clear there. You can see dark areas in the lungs of mice that were infected with the bug, with the wild-type bug, but not in the one that were infected with the mutant bacterium. So this opens up a lot of questions for us. This is again a survival curve. You can see on the left, this is just mortality being recorded. So the wild-type mice die within about 400 days, whereas you go up to 600 days, and those infected with the mutant bacterium are still quite healthy. On the right-hand side, you can see the loads of the bacteria in the lung. So the curve that's on the top shows you the wild-type and the complemented strains here. But this over here is the load of the mutant bacteria in mice infected. So you can see that it's not causing any symptoms of disease, but it still seems to linger on in the lungs of these infected mice. So this opens up a lot more questions for us. What's the difference between the wild type and the mutant? And does this shortening of a chain really have such a major effect? Is it just down to the mycolic acids, or is it just down to or is it more dependent on the whole ultrastructure of the bacterium? Is that changing? So this now brings us into a new area of research. So we now start looking at the immune response. What's the differences in the immune response to the wild type bacterium as compared to this mutant bacterium that can survive in the lungs but doesn't seem to cause any disease? So I'll leave it at that, and happy to take any questions.
I think we're very, very time, Luke. I think it's probably